Hi, my name is Erica. I'm a graduate student in the Gamillion Lab. I've been making videos on how to do cell culture protocols in case they don't exist in your lab or if you're brand new to the whole field. I'm making them to help teach my mentees and some students as well who are interested in cell culture. I've made videos already on how to make media and also about aliquoting. So now I'm going to be thawing cells today, which can be a little scary. It's a lot more involved than making media or aliquoting, but hopefully this video can help you get an idea of how you go about doing that, what's the safest way to do things. I'll discuss potential differences I've heard with other labs too, and how they do things, but I will be putting forth what I have personally learned to do in the Gamillion Lab. And it may not be the correct answer. You may be told to do something else, and you should probably follow that if you have good justification, if that is still maintaining aseptic technique and if it's good for the cells. So yeah, let's get started.
Hi, this is Erica from home. I'm inserting myself here to clarify that today, or in that protocol, I was thawing an adherent cell line. This is going to differ if you are working with a suspension-based cell line or culture, like CAR T cells, for example. It's going to further differ, maybe even within different adherent cell lines. You want to make the assessment for yourself to see if that particular cell line would be compatible with the protocol shown in that video today. If you are unsure about how to assess this, when you purchase a cell line, cell lines come with protocols typically, and if they don't, there's usually a lot of documentation out there on particular cell lines, or at least adjacently similar cell lines. In making these videos, I try to avoid going into all of the logistics about cell culture basics. If that's something that you're curious about or interested in, or you find you're questioning why cells even need to adhere in the first place, that's a whole other rabbit hole that potentially I make a video on in the future, but there's a lot of documentation out there already explaining those things. But I would like to point out sort of two topics that are relevant to this protocol and explain the steps a lot better than if I left it out. So the first term I want to introduce is passaging or subculturing. If you've not come across this before, this is the process of detaching and reseeding cells into new culture vessels to prevent overconfluency and maintain healthy growth. More broadly, it is the movement of cells from one vessel split into other vessels. And this is counted by an integer value that we refer to as the passage number. So this is how many times cells have been passaged since initial isolation or thaw. It's used as an indicator of cell age and phenotypic stability. So it's important to track passage number in order to determine how old your cells are getting. Eventually, they'll start to enter a phase called senescence, which is characterized by a decrease in differentiation potential and changes in, obviously, phenotypic stability, but also morphology and their ability to actually adhere on surfaces. When you're doing experiments, you want to typically work with a lower passage number because these cells are not aged yet and they're behaving as expected. So passage number is counted each time adherent cells are detached from a culture surface and reseeded into a new vessel. So in this image here, if we have passage 4 cells and we want to split them into a new vessel, this would become passage 5. Another way labs might be counting this progression of cell age is through PDL, which is population doubling level. This is a formula that takes an initial seeding density and final cell count and calculates a value that's associated with how old the cells would be. This is a more accurate count than passage number because it takes into account growth rate curves that are associated with cell culture. For the purposes of most labs, they don't track PDL, they use passage number, but this could be changing as time progresses. So I mentioned that in this video to say that when you thaw cells and they go into the flask should be labeled with their new passage number and not the passage number that they were frozen down at. Okay, now to actually talk about the thawing process. So cells are frozen or kept in liquid or vapor nitrogen. For HMSCs, it's more desirable that it is stored in vapor nitrogen, especially for the Rooster Bio line that I'm working with in the video. The temperatures of the cell bank are -200 Celsius. This is to halt biochemical reactions and to preserve the cells long term. As the cryovial is taken out of this bank and is brought into room temperature, it is so important to vent that vial. The large temperature gradient can form pressure inside the cryovial and cause it to explode in your hand, and you do not want that. So you vent it when you take the cryovial out. Another really important thing to note is that cells are frozen down typically with a cryoprotective agent. Most of the time, or at least what I have worked with and seen other labs work with, this is DMSO. And this is used to protect cells during freezing by preventing ice crystal formation. So it's really awesome, penetrates the cells and reduces intracellular ice formation. All of this to minimize osmotic shock and membrane damage during that process. However, when the cells are thawed and in their liquid state, it is a cytotoxic reagent to them. So once these cells are thawed, you need to move quickly to remove DMSO from the cells. This is why this protocol has a step in the center. Instead of just splitting what's in the cryovial into the flasks, we have to first separate the cells from the media containing DMSO, remove that, and then resuspend in fresh media before splitting into the two flasks. This is where the saying that everybody uses comes from: freeze slow, thaw fast. We want to thaw fast to get the cells out of the DMSO, but we want to freeze slow to prevent ice crystal formation.
First things first, go pee, because this is a long process. Wash your hands, come back, don your PPE and appropriate clothing, which is closed-toed shoes, long pants, a lab coat, safety goggles, gloves, sometimes a mask. If you're sick or coughing, you want to wear a mask. You don't want to cough into the biological safety cabinet. I'm going to heat up the things that I need for thawing cells, which is just media. I'm working with xeno-free media, and this is a situation where aseptic technique is extremely valuable in a lab like this. My biological safety cabinet is located in the same room as my benchtop work and other student offices, so super important that I have good technique, especially because this media is antibiotic free. Now I'm going to open the biological safety cabinet so I can turn on the light, and now I'm going to be waiting for laminar flow to be reached. I cannot work in the biological safety cabinet before that. I'm going to open my biohazardous trash and my sharps because I don't want to have to open it in the middle of the process. I know I'll be using it, and I don't want to be touching trash when I'm dealing, especially with antibiotic-free culture. So now that the hood has reached laminar flow, indicated by the green light, I'm going to wipe it down with ethanol. I'm spraying my hands very thoroughly with ethanol and rubbing them. In my biological safety cabinet, I keep an ethanol bottle to spray the bench top and wipe it down. My biological safety cabinet has a vacuum for aspiration. If it doesn't for you, you will have to manually aspirate. So in addition to the stuff that I'm about to add, you'll want to add a reservoir that has bleach in it for any trashed media if you need to do that. But because mine has a vacuum, we're going to also turn on the vacuum by fully opening the knob on the right. As I'm moving through the biological safety cabinet, I am very aware of my sleeves and where my arms are. I'm trying to stay on these armrests to prevent blocking the grates in the front of the biological safety cabinet, because that's where air is pulled into the system. If you block it too much, you're disrupting laminar flow. For cell culture today, I know I'll need a pipette aid. And this is just all stuff that's listed in the protocol. Before I even come in to work, I know the list in my head. And sometimes I actually write out in notes on the side of a protocol everything I'll need to place in the hood before starting. I know based on experience that I will just need 10 milliliter serological pipettes and 25 and 5 with this specific protocol. Just spray with ethanol too, and that's fine. And I place them on my clean side. It's because I'm right-handed, so I like to work with my right hand and have my working area here, and then my items more on this side. Today I'm following a Rooster Bio protocol for thawing, which calls for a 50 milliliter conical tube to have 10 milliliters of fresh xeno-free media sitting in it, waiting for the cells to be accepted into it, which is like pretty much our first step with handling the cells after thawing them. If you're in the Gamillion lab, this is typically a 15 milliliter conical tube with just 9 milliliters of fresh DMIM complete. So because I know I need a conical tube, I will get out 50 milliliter conical tube and a tube rack. And I'm planning on thawing into 4 T75 flasks, which I'm also going to get out and label with my initials, cell type, the date, and the passage or PDL numbered. So the bag I already have open just has 3 in it, so I need to get out one more fresh bag to open. Now, you may want to thaw in a different way, or your protocol for your specific cell line may require something else. This is just what I'm doing for today and what I know I need because I'm growing out a cell bank. Okay, so now I'm going to take the flasks out and I'm going to label each one with what I'm about to thaw into them. You open fresh bags like these that are sterile, like a chip bag. I only need one out of here. So I'm going to shimmy one out because you never want to reach into a bag. This is a canted neck T75 flask, rectangular with a filtered cap on top. So you don't want to ever touch that filter there, that white dot. That's how gas exchange occurs within this flask. This is also plasma treated. So I'm not going to be coating today. It already has what I need for the cells to adhere to it. And the media already has what that needs to pertaining to that. I place them towards the back of the biological safety cabinet, filter side facing away from where dirty air might be coming in. Potentially, shouldn't be, but. And then again, I'm folding this like a chip bag where I take the corners in and then roll it down. These flasks come with a little piece of like tape to use. I don't ever use that because I find that it's not strong enough to actually keep the bag closed. So I hold this with lab tape. Okay. Now this has the other 3 flasks that I need. I don't remember if I already addressed that in this video, but in my lab we put all non-biohazardous trash just thrown behind us to get it out of the way, because we don't keep such a trash can next to the biological safety cabinet, because you never know what's growing in a typical trash can. At least with the biohazardous trash, there's bleach and stuff in there, like things have been hopefully decontaminated before thrown away in there. I'm gonna get out a 50 milliliter conical tube as well. I'll remove these out now because I no longer need them. And now I'm going to label my flasks. When I'm labeling, I'm making sure my bare skin isn't touching the flask. Okay, and some other things for setup is that I'm going to turn on the centrifuge and go ahead and counterbalance it with the volume that I know to counterbalance it with based on what I'm going to have as a final volume in that conical tube today. So I'm turning it on, opening it, and just from experience, I know the final volume for this situation is going to be 12. Okay, I'm going to get my media from the water bath. I put a piece of paper towel down. My media in a bottle this large takes like 30 minutes to thoroughly heat up to the temperature that you want, 37 degrees Celsius. I'm going to wipe off starting from the cap and then drag away from the cap so that I'm not pushing potential contaminant into the cap. Wipe off the bottom, and then I'm going to spray it with ethanol and add it to the hood. Because of the freeze, slow, thaw, fast acronym I talked about earlier, I'm going to go ahead and put fresh media into my conical tube as well, so that in this next step, when I get out my cryo vial and I thaw it, I can immediately put that thawed content into the conical tube and then start spinning it down in the centrifuge. So always open containers. Top of the cap facing down, away from you so you're not hovering over it. I need 12 milliliters of media. I can use a 10 milliliter serological pipette and overfill it to the 12 mark. I'm opening it as intended and not tearing through the paper to maintain sterility. Open it just a little bit at the top on where the cotton side is. Attach it to your pipette aid and face the numbers towards you. You can move it around when the paper's still on. Once this paper comes off, you can't touch this serological pipette anymore or it won't be sterile. So I like it on this side. I'm gonna draw up 12 milliliters and add it to the conical tube. I'm trashing this pipette. And I'm gonna go ahead and cap it because I'm temporarily not using it anymore. And now I'm ready to go get my cells. I'm also going to double glove so that when I'm manipulating the box, once it's outside of the cell bank, I don't get freeze burn on my fingertips and stuff. So I'm going to put on another layer of gloves on top of this. And I put on cryo gloves too. You want to always wear these if you're reaching into this tank. This may vary a little bit in your lab, but with my non-dominant hand, I'm going to open the lid and then reach inside for the color rack that I want. So in my lab, we've labeled these racks with colors. If you look closer, sometimes there's numbers next to the rack. I know that I need to get yellow. So with my dominant hand, I grab yellow. I'm bringing it to the center so that I can lift it out. And you want to stand back while you're lifting it out. And I like to let the liquid nitrogen drain out of the boxes before fully pulling it out. If you don't try to drain as much as possible, you risk liquid nitrogen being in the tops of the cryo vials when you grab them, and it's not very comfy to hold it when it's that cold. Okay, I'm gonna bring the rack over here, place it down, re-lid the cell bank while I'm manipulating with that so I'm not wasting liquid nitrogen evaporating out of there. And I actually need box 1, so I'm gonna grab this piece to unlock it, tap the back of the rack to get the box out, and because I have 2 layers of gloves on, this does not hurt. There's little indentations here on the side of the box so you can grab to pry the lid off. And I'm getting out my cryo vial here. Immediately you want to vent it. This one wasn't very pressurized because it's new, but sometimes they get pressurized with the temperature change. You want to verify that you pulled out the right one. I did. I'm going to put this box back, and I'm going to bring this to the water bath, put it in one of our styrofoam floaters, and let it thaw. Now, it does not take long at all to thaw the cells, so I'm going to quickly relock the box, fully locking it, putting this part in its hole. There. Put back on my gloves. And return this rack to the cell bank. Now I'm going to put this back where it goes. So I'm going to check on this cryo vial now. As soon as you see that the contents are able to slide, as soon as you're able to see that the contents inside are able to slide into the lid, then you can bring it to the biological safety cabinet to put it in the fresh media. Sometimes I will sit here and kind of move it through the water bath water to encourage thawing. Okay, so now when I tilt it, the contents inside are falling to the lid, which means that it's thawed enough to now take it to the biological safety cabinet. Now that it's liquid, I'm moving quickly. Normally I would hold this in a tube rack, but the Rooster Bio cryo vials don't work with the tube racks that we have in our lab currently. So I'm being very careful as to not knock that. This I adjust to 1000. I'm carefully opening, not messing with any of the other tips, and picking up the tip that I need out of there, like so. I'm turning the vial towards me so that I can see kind of inside of it. And I'm going to pick up all of the contents and put it all into my media. When I take off tips, I hit the button of the micropipette and take off the tip in the biological safety cabinet and then throw it away. I am not bringing the micropipette outside of the hood to then trash a tip. I'm gonna keep the micropipettes inside the hood. I'm gonna bleach this vial because I'm done with it now. And optionally, and what I'm going to do is keep the vial in the hood until I'm completely done thawing and putting the cells in my flasks to make sure that I'm labeling everything correctly. So now this final volume's at 12 milliliters. I have a counterbalance already in the centrifuge. And at this point you can relax a little because the cells are now being diluted a little bit with fresh media in DMSO. Make sure this is screwed on well before centrifuging, and then go ahead and start that. So I close that, and actually what this protocol calls for is for this to be run at 400 G. So I'm going to start that, and it's got to be done for 10 minutes. So usually I wait and I kind of feel the side to make sure that it's started and it's not out of balance, so this is good. So now I have 10 minutes to kind of prep for the next step. I'm going to check off in my cell bank that I just took out that vial before I forget. And I'm going to fill my flasks with some media. Once those are spent down, I'm gonna have media that has DMSO and a cell pellet. I want the cell pellet, I don't want the media. So I'm gonna aspirate out that media, leaving the cell pellet, resuspend in 5 milliliters, because 5 milliliters, no matter what you want your ending volume to be. The 5 milliliter serological pipette, I'm gonna open one just to show you. But the 5 milliliter serological pipette is what you always want to resuspend in. It has the smallest nozzle exit diameter, and it will help break up your cells fast. You don't want to spend too much time breaking up your cells because you can traumatize them. So I'm resuspending that cell pellet in 5 milliliters of fresh xeno-free media. Then I'm splitting it 4 ways into the 4 T75 flasks. This will make it so that I have a cell suspension in each flask. That is 5 divided by 4, which is 1.25 milliliters that will be in each flask. However, I know that the working volume of a flask must be at 10 milliliters, typically in the Gamillion lab. So I need to add that much media to each flask to get it to 10 milliliters. So 10 milliliters minus 1.25 is 8.75 milliliters of fresh media that I could add to the flask right now in order to prep for when the cells enter. And then they're just ready to put in the incubator. With Rooster Bio, it is recommended to work with a working volume of 15 in there. So I can add 15 minus the 1.25 cell suspension that I'm expecting to be put in there to each flask. So just to re-summarize, 1.25 milliliters of cell suspension per flask, and then I'm supplementing with 13.75 milliliters of extra fresh media to have 15 milliliters total in each flask. Now, 2 things. One, Rooster Bio recommends to currently use cell-bind flasks. I'm just using the flasks that are available in my lab because it's fine. Second thing, when I'm actively working with a flask, I work with it sitting up, with the filter facing up, so I can open and close the cap and put things into it. When it's in the incubator, it's laying down correctly so that the cells adhere to the correct side and get their media. This may differ from your lab. Loosen all the caps of the flask. I'm not touching the filter on the top because that could compromise gas exchange. Opening my media bottle. And what I'm about to add is the fresh media, which is 13.75. So I will need the 25 milliliter serological pipette to even hold that much. When working with a 25 milliliter serological pipette, it's prone to dripping from its nozzle, so just be very slow and moving when you're using it. And anytime you add liquid to a flask, you want to be working in that corner as close as possible, especially when cells are in there. But I think it's a good idea to just do it always so you never forget. Okay, so I'm going to trash this serological pipette. Recap my media for now. And I've not tightened those caps yet, but I do set them back onto the flask just to protect the contents of the flask. Okay, I've turned off the centrifuge, put the counterbalance back, and I've also turned off the water bath because I don't need those things anymore. This is the results. There's a very small cell pellet at the bottom. It's hard to see on video. but trust me, it's easier to see when there's phenol red media in there. But now it's ready to remove this dirty media that has DMSO in it and resuspend in 5 milliliters fresh media. Spraying this liberally because it was just in a dirty centrifuge. Spray my hands. Always loosen the caps first. I am going to get a glass pasture pipette, open the lid, put it out the way, and I'm going to grab the glass pasture pipette at the very tip and be careful not to touch any of the other ones as much as possible, because everybody in my lab, everybody uses the same glass pasture pipette. I attach this to a vacuum, remove the lid, turning cap away from me, not towards the palm, and then I hold the conical tube like this. I aspirate any remaining bubbles if there's any, but there's none here, and then I remove the liquid, leaving the cell pellet at the bottom. Return the conical lid back to its position. Remove this glass pasture pipette, and then this goes into the sharps. If you don't have a vacuum, you might have to manually aspirate that and put it into your reservoir that you have in the biological safety cabinet. Next, I'm going to get my media, get a 5 milliliter serological pipette, open it just at the tip, attach it, turn it so the numbers are facing me, draw up 5 milliliters, turning the lid, and this is probably a very difficult thing to do potentially, but you're going to resuspend this cell pellet without making any bubbles. So this is a skill that you may need to practice. It's sort of bouncing up and down, and I still make bubbles sometimes between 3 and 2 until you see that everything is broken up, which mine is. It may take more than that. I'm gonna return the cap, and actually I'm gonna go get 2 milliliter serological pipettes. Add some of those. And this will allow me to measure the 1.25 cell suspension that should go into each flask. And if I'm careful and I don't hit this tip on anything, I could use the same serological pipette to seed all of my flasks. Yeah, 1.25. Seed into the corner, like so. Close that flask. And move it away so I know that I've already seeded it. And I'm being very vigilant to make sure that I'm not dripping off of the serological pipette. Now you're not done yet. This is possibly 2 of the most important steps. Make sure that all the caps are tight. And now you want to rock the cell suspension on the back of the flask so that the water tension is broken, and it can actually like reach and coat all of that rectangular space that's on the back of the flask. Okay, closing the media. And now I'm going to take these and put these in the incubator. But first I'm going to check on the microscope if cells actually made it inside the flask. It's hard to tell or confirm seeding density at this point. but based on what I'm seeing cells floating around in media, so it's likely that I did seed correctly. I'm going to check every single flask. Every flask was good. Now I'm going to spray every flask with ethanol and put it in the incubator. You want to treat the incubator like a biological safety cabinet as well. I don't want to introduce any contamination in there either, because that would be really bad for the lab. I'm placing filter to the back. In industry, you might see people do otherwise, but it's the technically right way to prevent contamination. One, because the filter's not facing outside dirty air for as much as possible. But also when I go to grab it next time, I'm not grabbing it by the filter side. I can grab it by the bottom and then bring it back to wherever I need to work with it. I'm holding it, pinching the bottom, spraying on all sides. And you never want to stack higher than 3 because it can start to get precarious or affect temperature gradient. So this 4th one, I'm putting next to the 3 that I just stacked there. I turned off the microscope. I plugged up the pipette aid back on the charger. I'm going to put all of these things away, still spraying with ethanol on my hands to put them away, and then I'm going to wipe down the bench top in there. The conical tube gets bleached because cells were in there and thrown away. Again, leaving the lid a little loose so that you're not putting a potentially pressurized conical tube in the autoclave. Grabbing this stuff. I no longer need the vial. I put bleach in it earlier. And take these out. Because I used the reservoir this time, I need to flush my vacuum. If you don't have a vacuum, you don't have to worry about this, with ethanol. So it should still be on at this point. And pulling a little bit of ethanol in. Sometimes I like to plug this hole to kind of get it to travel all the way through in the Gamillion Lab vacuum. Recap this. This has been wiped down. I turn off the vacuum. Close the sash. UV light it because no one's going after me for further sterilization of the inside. And now I'm going to empty out the traps. Sometimes both need to be emptied out down here. We have 2 in the Gamillion Lab, but just this one seems to need emptying out. Want to turn on the water and do it a specific way. And I am allowing the water to run and help dilute this as it goes down the sink. And I leave the water running while I refill it with bleach located here. Enough to restore what was at the bottom. This is important to decontaminate any aspirate you might have from your work. Then I come over here and turn off the sink, and now I'm going to re-plug this back up. Make sure that you fully return the plug to this flask and connect this connector. Otherwise, it'll have a weak pull for the next person. And finally, pick up any trash off the floor if you operate like the Gamillion Lab does, to make sure that's clean, and recap the biohazard trash can so that you are compliant to the regulations of having a biohazardous trash can. Picked up these off the floor, and this can go in regular trash.
So now you have active cells growing out, and it's important that you come in every day to check on those cells. If the cells are growing out for more than 3 days, then you should be changing media. Media should be changed every 2 to 3 days for cells in active culture. If more media needs to be made, pay attention to that. media is only good for about 4 to 6 weeks, and then you need to make new media. Checking every day is important to ensure that contamination hasn't occurred. If it has, you can intercept that immediately. If your cells have died, you can quickly thaw more cells and not delay an experiment too much. But more importantly, you can monitor when they have reached 90% confluency, which could look something like this. At that point, you would want to do something with the cells, passage them likely, and I will be making a video on that. So thank you for watching.